The last topic to be covered for this pre-exam review is muscle. And as you remember, as you all know, I'm sure, there are three different types of muscle in the mammalian body. Skeletal muscle, which is under voluntary control, and it can generate rapid movements by sudden bursts of contraction, such as needed for walking, running, writing, etc. Cardiac muscle, which resembles skeletal muscle in many respects, but is specialized for the continuous involuntary contraction needed for the pumping of blood. And smooth muscle, which is also under involuntary control, which surrounds internal organs such as the intestine, uterus, blood vessels. And smooth muscle can contract slowly but can create and maintain tension for long periods of time. Since most studies, the most complete studies, have been done on skeletal muscle, we'll look at skeletal muscle in some detail. A named skeletal muscle, such as the biceps, the triceps, is wrapped around by a layer of dense irregular connective tissue called the epimysium, and this epimysium defines the total muscle. The total muscle is made up of a number of fascicles, and fascicles are bundles of muscle fibers or muscle cells, and they are wrapped around by a layer of connective tissue, also dense irregular connective tissue called the perimysium. And the perimysium is the pathway for the entry of blood vessels and nerves into a muscle. Each fascicle consists of many individual muscle fibers, and these muscle fibers are wrapped around by a layer of very loose connective tissue called the endomysium. And then each muscle fiber or muscle cell is surrounded by a plasma membrane or sarcolemma and contains sarcoplasms, the sarcoplasm, and many myofibrils, which we'll look at in some detail. The myofibrils, shown here within a myofiber, are just a long series of sarcomeres joined together and separated by Z-discs, and we'll look at the structure of the sarcomeres in some detail. Now, muscles are richly innervated and have a rich blood supply, and a nerve bundle, before it makes contact with the muscle, breaks up into a number of branches, and these branches then make contact with individual muscle fibers. Here's a muscle fiber, actually labeled S here for striated muscle fiber. Here's a muscle fiber, and the contact that the terminal branch of a nerve fiber makes with is called a motor end plate. And all of the muscle fibers that a single nerve bundle contacts is known as a motor unit. So all of these motor end plates, which are derived from this nerve bundle, constitute a motor unit. Skeletal muscle fibers are multinucleate, and they formed by the fusion of many hundreds of myoblasts forming a multinucleate myotube. These myotubes then manufacture the myofibrils, the specialized myofibrils that fill each muscle cell. And as the myofibrils are synthesized and fill up a myofiber, they take up pretty much the entire content of the muscle fiber, and so all of the organelles such as the nuclei, mitochondria, the endoplasmic reticulum are pushed to the side of the muscle fiber and take up residence right under the plasma membrane or sarcolemma. The number of muscle fibers within a muscle doesn't increase much after birth. What does increase, and what we can increase by weightlifting and by exercise, is the formation of more myofibrils within a myofiber. So since we can't increase the number of or type of muscle fibers after birth, if you want to be an Olympic athlete, you better pick your parents very carefully. Muscles contain 3 different types of fibers, and a single muscle will contain all of these different types of fibers, but may contain more of one type than the other. Red fibers, also called S here, red fibers are red because they contain large amounts of myoglobin, which is the oxygen carrying protein of muscles, large numbers of mitochondria, and they rely on oxidative phosphorylation for the production of ATP. Since oxidative phosphorylation is a very efficient way of producing ATP, these muscles are slow twitching, they always have a good supply of ATP, and they're slow to fatigue, and are typically found in the limb muscles of mammals, in the flight muscles of birds, and in the muscles of long-distance runners. White fibers are larger, contain less myoglobin and few mitochondria, and they are high in glycogen, and they derive their ATP from anaerobic glycolysis, which, as you recall from your biochemistry, is much less efficient in producing ATP than is aerobic oxidation. They are fast twitching, and they fatigue easily due to the buildup of lactic acid and oxygen debt, and they're used to control fine movements such as movements of the fingers, etc. And sprinters have lots of white fibers in their muscles, and then intermediate between white and red are the intermediate fibers, fall somewhere in between in mitochondria, size, twitch rate, etc. The basic unit of contraction is the sarcomere, and the sarcomere is the distance between 2 Z bands. It consists of a light band, a dark band, and another light band. The light band actually stretches between the 2 dark bands and is bisected by this darker line here called the Z disc. A sarcomere is about 2.5 microns in length, and it's shortening of the sarcomeres which results in contraction of a muscle. Sarcomere contains 2 main types of proteins, thick myosin filaments and thin actin filaments. The thin actin filaments are inserted at one end into the Z disc, and they partially project into the myosin containing part of the sarcomere. It's the part of the sarcomere that contains myosin is known as the A band or dark band, and the part of the sarcomere that doesn't contain myosin is known as the I band or light band, and that is the band that is bisected by the Z disc. In addition to actin and myosin, which are the contractile filaments, there are many other types of proteins which maintain the exquisite array and organization of these 2 contractile filaments.